In previous module, you learned about mechanism of phytate hydrolysis in the gut and absorption, and also learned its physiological effects of phytates. In this module, you are going to know about the beneficial effects of phytates. For decades, phytate has been regarded as an anti-nutrient, as during gastrointestinal passage, it may inhibit the absorption of some essential trace elements and minerals, which under certain dietary circumstances may lead to calcium, iron and zinc deficiencies. Thus, intensive research has been carried out to remove phytate from food by proper processing to improve the bioavailability and to avoid deficiencies of essential trace elements and minerals. In the last 20 years, however, beneficial properties of phytates have been observed and antioxidant and anti-cancer activities were reported. Inhibition of calcium salt crystallization and prevention of renal stone formation through dietary phytate were described. Reduction of starch digestion along with slowing down of glycemic index of foods have also been reported, as well as positive effects on blood glucose and blood cholesterol. In this module, we shall study the beneficial effects of phytates. Phytic acid is a known antioxidant. Colonic bacteria produce oxygen radicals in appreciable amounts. Dietary phytic acid suppresses free radical mediated damage to the intestinal epithelium. For example, because phytates can chelate copper, iron, zinc, magnesium, manganese and several other minerals, and many of these minerals are essential cofactors for numerous oxidoreductases. Several investigators have suggested that phytates may act as an antioxidant in vivo. Such antioxidant activity has been suggested to be responsible for free radical scavenging action in vivo that may lead to anti-cancer activity. Mineral chelating ability of phytates has also been suggested to be responsible for antimicrobial like mold inhibition, for example, activity. Protective benefits are seen particularly in relation to carcinogenesis, particularly against the colon cancers. Complexing of ion catalyzed production of free radicals and reduce lipid peroxidation, lower blood glucose and lipids, binding amylase reducing starch digestion, lowering plasma cholesterol and triglycerides. In the figure, you can observe possible mechanism of phytic acid on protein binding, starch binding and mineral binding as well. Now, we will discuss perceived effects of phytic acid in risk reduction of diseases. Reduction in cancer risk. Different cancers, especially colon and breast cancer, has been linked to phytates. Endogenous phytic acid derived from high dietary fiber foods such as wheat bran along with exogenous pure phytic acid added to a low fiber diet may be colon cancer protective. Phytic acid consumption as a part of diet may potentially lead to starch malabsorption, resulting in colon content dilution, increased butyrate and decreased pH, which could be potentially protective of colon from carcinogenesis. Also, due to its structure, phytic acid can chelate polyvalent cations such as iron, calcium and zinc. These minerals are required for vital cellular functions and their chelation may affect mineral vital to DNA synthesis and cell growth may be a factor in studies that have shown in decrease in cell proliferation with phytic acid supplementation. Phytic acid may also exert its effects by affecting the immune system. Enhancement of natural killer cell activity has been shown in carcinogen treated mice fed with phytic acid and in vitro when splenocytes from normal mice were treated with phytic acid. Purified phytic acid has been shown in vitro studies to inhibit cell growth and increase cell differentiation and maturation of HT29 human colon carcinoma cells. Learners, let us see the effect of phytic acid on heart disease. A high zinc copper ratio has been linked to hypercholesterolemia. Thus, by virtue of its ability to bind these divalent cations and potentially alter their balance and availability, phytic acid may also affect serum cholesterol levels. The ability of phytic acid to chelate iron and possibly reduce its free radical generating potential and subsequent lipid peroxidative damage may also protect the heart from ischemic and reperfusion injury. It has also been suggested that the calcium binding ability of phytic acid may help to reduce the level of calcium in soft tissue prone to calcification. Seeley suggested that calcification of aorta may lead to loss of elasticity and development of hypertension. It is argued that phytic acid may bind excess calcium, reduce the level of aortic calcification and improve hypertension. Now, we will go through hypoglycemic effects of phytic acid. Of the different carbohydrate foods tested for starch digestibility and blood glucose response, the rich in anti-nutrients, example legumes, show the lowest values. Amylase inhibitors can reduce blood glucose and raise insulin levels. Phytic acid may play an important role in the inhibition of starch digestion and absorption. It can bind to starch either by hydrogen bonding, indirectly via proteins it is bound to, or by inhibiting the proper digestion and hence absorption of starch by binding amylase or enzyme cofactors such as Ca2+. Any starch that is not digested and absorbed in the small intestine would probably lessen the glucose absorption at that site, thus decreasing the glycemic effect and the required insulin response. This is of consequence not only in diabetes, but also in heart disease, because reductions in plasma glucose and insulin may lead to lower hepatic lipid synthesis. The hypoglycemic effects of phytic acid may also contribute to its hypolipidemic effects described above. The final topic of this module is effect of phytic acid on other diseases. By virtue of its mineral binding abilities, phytic acid may also aid in the prevention of renal calculi. Phytic acid has been shown in vitro to best prevent bruchite, a form of calcium phosphate and component of renal calculi crystallization and precipitation. Calcium and fluoride binding by phytic acid can insolubilize the minerals and therefore may be useful in the development of dental enamels. Phytate and sialolithiasis. Sialolithiasis is a common disease of salivary glands. Little is known about etiology of these calculi and their exact mechanism of formation is unknown. It was concluded that a deficit of crystallization inhibitors such as phytate is also an important etiological factor of zeolith development. Phytic acid also forms an iron chelate that suppresses lipid peroxidation by blocking iron driven hydroxyl radical generation. The primary mechanism by which the beneficial health effects of phytates may be explained is the strong mineral chelating ability of phytates. Calcium and fluoride binding by phytic acid can insolubilize the minerals and therefore may be useful in the development of dental enamels. Let us look into a conclusion of the subcontents of the phytates. Phytate is a food component that is considered an anti-nutrient by virtue of its ability to chelate divalent minerals and prevent their bioavailability. Thus, intensive research has been carried out to remove phytates from food by proper processing to improve the bioavailability and to avoid deficiencies of essential trace elements and minerals. Studies showed inhibition of calcium salt crystallization and prevention of renal stone formation through dietary phytates. Reduction of starch digestion along with slowing down of glycemic index of foods have also been reported, as well as positive effects on blood glucose and blood cholesterol were also shown. The phytate concentration in the diet has to exceed a certain level to have a substantial effect on bioavailability of minerals and trace elements. In healthy individuals taking balanced diet, the effect of phytic acid on inhibition of mineral absorption is minimal and does not cause any nutrient deficiencies. Lastly, to reduce the anti-nutrient effects of phytic acid in foods, the procedures like soaking, sprouting, fermentation and consuming vitamin C rich food enhance mineral absorption and offset phytic acid. Thank you.